Hey everybody, I want to push out these epigenetics notes sooner rather than later because several of you expressed confusion in epigenetics. So let's talk about it and kind of wrap it up. You've already read chapter 11 in your book. You should have done some research on epigenetics, and so hopefully this will wrap it up nice and neat. So let's start with just defining what it is. If genetics is the study of your DNA and your heritable features, or your nature, so to speak, then epigenetics would be your nurture. And so, how does the environment affect the expression of your genes? Um, and so, a more formal definition, it is the study of DNA modifications that involve mechanisms that don't change DNA sequence but do affect gene activity and alter phenotypes. And so, epigenetic changes will result in differential gene expression, increasing the expression of some genes and silencing or repressing the expression of other genes. And that's in contrast to gene mutations, which actually change the base sequence of the DNA. This does not irreversibly alter DNA. Mutations um, are irreversible, but epigenetic changes are reversible. Some of them are not easily reversed, but they are reversible nonetheless. So let's review chromatin structure a little bit. We talked about the cell cycle and that during mitosis, that's when chromosomes condense. But most cells, most of the time, are in interphase of the cell cycle where the DNA exists as chromatin, which is less condensed than chromosomes. Chromatin is a complex of both DNA and histone proteins. And so the DNA wraps around the histone proteins, and that kind of condenses everything so that it can be bundled and fit into the nucleus. The structures that are formed from the association of the histone protein complexes and DNA are called nucleosomes. And even within the chromatin, the degree of coiling is different. And so the more coiled form of chromatin is called heterochromatin, and it's coiled tightly enough that it is inaccessible for transcription. And so the transcription factors and other proteins needed for transcription cannot access the base sequences when it is in the form of heterochromatin. In the form of euchromatin, it's a little bit more diffuse and less compact, and it can be transcribed. So there's three epigenetic mechanisms that we're going to go over. The first one is DNA methylation, and that one is probably the most interesting, I think, because it is the one that is really kind of influenced by environment. And so there's been studies, and if you explored those HHMI pages that I had you do, you would have read about some of the twin studies that have been done and studies in mice um, that show how methylation um, affects phenotype over the life of an organism. And then the other one is histone modifications, so also chemical modifications, but to the histone proteins instead of the DNA, and microRNAs, which is a form of post-translational control. Um, that's all I'll say about that for now. So, starting with DNA methylation, a methyl group is just a carbon with three hydrogens attached to it, and so DNA methylation is the attaching methyl groups to specific nucleotides, usually cytosine residues in the DNA sequence, and it down regulates transcription. And so, an area that of the genome that is highly methylated is not going to be transcribed. The regions in the DNA that tend to be methylated are these CPG regions. In other words, just areas where cytosine and guanine residues are side by side on a DNA strand. So the P in there just stands for the phosphate that would be in between the two bases on the DNA strand. And about between 1 and 5% of cytosine residues in your genome would be methylated by the end of your lifetime. And so DNA methyltransferase is an enzyme that will attach methyl groups to the cytosine residues. When the DNA is replicated, the nucleotide sequence is copied, but not the methyl groups. Methyl groups are added, though, retroactively by another enzyme called methyltransferase. And so if a specific residue is methylated, then when it is copied, the methyl group ends up essentially being copied with it, even though it's not copied during DNA replication. And these cause long-term changes in gene expression. So again, the methylation occurs over the course of a lifetime and accumulates, and there have been studies to show that they can be chemically removed, and perhaps by behavior changes as well. But generally, they're semi-permanent. However, when fertilization occurs and there's a new embryo, the slate gets wiped clean and is demethylated. And that is an important process in embryonic development. I'm going to show you one specific example that is where methylation is important, and that is important for development. And that example is X-Inactivation. So I don't think we talked about this too much during genetics. We might have mentioned it, but X-Inactivation is what happens when a female who is XX is in the embryonic stage of development, and each cell has 2 active X chromosomes. What happens is that one of those X chromosomes in each cell will get deactivated, and it gets deactivated randomly. And so any particular cell may have, after inactivation, may have a maternally inherited active X chromosome or a paternally inherited active X chromosome. And the other inactive chromosome, the one that has become deactivated or inactivated, becomes what's known as a bar body. The reason this happens is so that in males and females, essentially what you have is, whether male or female, there's still only just one active X chromosome. And so it ensures that the X-linked genes are expressed kind of in equal amounts in males as well as females. The consequence of that is that all of the somatic cells that are derived from those embryonic cells have either a maternally inherited or a paternally inherited X chromosome, and those cells divide and form tissues, and those tissues end up forming like a patchwork. And so women are a patchwork of tissue where there are alternating maternally and paternally inherited X genes being expressed in those tissues. And the most common and most visible example of that is in calico cats. And so they've got that patchy fur color, and the patch is the result of X activation. So the pigment is an X-linked allele, and in calico cats, they are heterozygous. And so some patches are light colored because the X chromosome from one parent is being expressed, and some patches are dark colored because the X chromosome from the other parent is being expressed. Histone acetylation is another epigenetic mechanism. It upregulates transcription and so promotes transcription of genes in those areas. And so if this is a nucleosome, you've got your DNA and your histone complex. The histone proteins have um, these little tails that are positively charged because they contain lysine residues. Lysine is an amino acid, whoops, go back, that is positively charged. And so it is attracted to the negatively charged DNA and they form kind of a tight association. When the lysine residues are acetylated, that positive charge is neutralized and that weakens the attraction to the DNA. And so the histone tails kind of loosen up, the DNA unwinds a little bit, and everything becomes more accessible for transcription factors and regulatory proteins. So transcription can occur. And these are, if the DNA methylation is affects long-term changes, these are kind of short-term changes in gene expression. And then finally, microRNA. This is just showing gene expression normally. MicroRNA are just small pieces of RNA and siRNA as well. They're very similar that will have complementary sequences to mRNA and they will bind. And when they bind, they either block the translation of the protein or in some cases degrade the messenger RNA. So that is that's epigenetic mechanisms in a nutshell. I'm going to post a article on Google Classroom on epigenetics that's kind of easily readable and pretty interesting and specifically mostly about DNA methylation. And so I encourage you to read that. And I'll leave you with this. And we'll see you on Monday.